So now we're ready to talk a little bit more detailed about protein synthesis. Now, protein synthesis starts with DNA, and ultimately we get mRNA. And this process of taking taking DNA and converting it into mRNA is going to be called transcription. And because that DNA molecule is located in the nucleus, this process of transcription occurs at the nucleus. mRNA is able to leave. It's small enough to fit through those nuclear pores, right? And so it's going to actually go out to the ribosome, and it's going to have the instructions. Remember, it's the recipe for building that protein. And this process of going from the mRNA to the protein is called translation. And this is, of course, happening at the ribosome because the ribosome is where protein synthesis occurs. Now, it's kind of unfortunate that these words are so close to each other. So we have transcription and then we have translation. And if you pay attention, you know, a good memorization technique is to use the alphabet when you can. And so you may be able to see that the first letter that's different in these two words is C and L, and C comes before L in the alphabet. And so maybe use that to remember that transcription occurs before translation. Me personally, I always say it transcription, translation, transcription, translation, so that it sounds correct. And if I go translation, transcription, I'm like, oh, there's something off there. That sounds wrong to me, right? And so I pick up on that really fast. So that's part of the training that I did as I went through this. Okay. There's a couple other terms I need to introduce before we get into the details of this process. So I actually want to go back to our picture here, our slide on DNA. And I want to define the term gene. So a gene is actually composed of DNA. It's a certain section of the DNA molecule that codes for a protein. So I would like to think about the term genome. That's going to be all your genes as maybe the whole cookbook, right? Maybe it's even like grandma's cookbook. So it's like really sacred. It's really special, right? You want to protect it. It's got all this family heritage, right? It's got grandma's recipes. And then the gene would be like, let's just say, an individual recipe. This is just like one dish. This is just like one dish. Recipe for one dish would be the gene. This is our functional section of DNA, right? Is the gene. So when we're talking about making a protein, we're talking about making a protein. And we're talking about taking that DNA and converting it into mRNA. We're talking about doing this on an individual gene level. So we're basically taking not the entire genome, but we're just taking a single gene and we're converting that gene from DNA to mRNA. And what I like to think about here is the function of that mRNA is, again, it can leave the nucleus. So this is like this ability to take grandma's recipe, your favorite dish that she made for you that was special, and then take that recipe card out of her kitchen So maybe in the old days, you had to get a little index card and you could read from her cookbook and you would write down her directions on your index card or your piece of paper, right? And you would take it. Now maybe you just snap a photograph with your phone, and so you still have grandma's recipe and it's sacred to you, right? And maybe that's even more special because you get to see her handwriting perhaps, or maybe it's grandpa's recipe, right? It could be anybody's recipe here. And so the mRNA is this ability to take that sacred recipe and allow it to leave grandma's kitchen. My grandpa didn't cook, so I'm going to keep saying grandma as in my own personal family history, right? And maybe your family history is different. That's okay. Okay. And so when we look at that mRNA, it has this ability to leave that nuclear envelope. It can move through that pore because one, it's not the entire genome, it's just one gene, it's just one recipe card. And two, it's single-stranded rather than double-stranded. So it's a significantly smaller molecule. And so it's able to leave and go to this kitchen, go to this ribosome where we're going to make our protein. And so another thing that we can do here is represent if we wanted our nuclear envelope, and we could show like our nuclear pore, and then this line here could be like our nuclear envelope. And notice that we have the mRNA able to fit through that pore. So this is the significance of, again, that mRNA. Another way that we can talk about that mRNA is we can call it an mRNA transcript, where it's this product of transcription. And the beauty of the mRNA is that it's short-lived. So it's going to go out to the cell. It's going to be used at the ribosome to build that DNA molecule, and then it's going to break down. So if there's any mistakes in the mRNA, it's short-lived. It won't matter for a long period of time. Okay, so let's go back to this picture originally where we drew like a section of the nucleus and we do our nuclear envelope and we have our nuclear pores. And inside, let's go ahead and draw out for ourselves. I'm making my screen really huge. I'm hoping to have plenty of room to draw this out. I'm going to draw my DNA molecule. So remember, my DNA molecule is a double helix, so I'm going to draw it in a vertical direction. And then what we're going to see happening is it has to open up and unwind a little bit so that we can come in and do our process of transcription. And so let me go ahead and also in my DNA write out my nitrogenous bases. So I'm going to do oops. I'm going to do A, T, G, A, G, G, T, A, T, T, A, G, C, C on one strand. And then I'm going to write the complementary sequence on the other. T, A, C, T, C, C, A, T, T, and I apologize if my drawing isn't lining up exactly across. It's doing a pretty good job. So on my complementary strand, I have starting again TAC, TCC, ATT, AAT, and finishing with CGG. So this is my DNA, and I'm using black ink here. And then in order to represent mRNA, I'm going to switch colors to a blue, and I'm going to write out my mRNA transcript. And the only secret here is anytime I see an adenine in A, I'm going to pair a U with that. And anywhere I see an A, I'm going to pair a T. And then C's and G's stay the same. So I'm going to do the top sequence. I'm going to turn that into mRNA transcript. So I start out with UAC, UCC, AUU, AAU, CGG. And I kept looking at my notes when I could have just done the transcription. And so this is my mRNA transcript. And then I'm going to make my screen small again. Okay, and just a couple notes. What we're looking at here, this is our first step in protein synthesis. We call it, again, transcription. This is involving some hormones. So again, we have RNA polymerase this time, which is going to unwind and build the RNA strand, the mRNA. Now remember, in our RNA strand, we have single-stranded, we have ribose instead of deoxyribose, and we have uracil instead of thymine. Now what we're going to see happening is that while this mRNA is still in the nucleus, we're going to see that the mRNA is going to be edited before it leaves. And so we have an enzyme called spliceosome, which is going to cut out and splice cut out or splice. That's what I meant to say. So the term splice here means to cut out sections. And we call these sections that get cut out introns. And then we'll see it's going to paste remaining pieces together. Those would be called exons. And what we see then is this editing can allow us to use the same gene and cut and paste it in different ways and actually use that to make different proteins. So this is pretty cool. So the spliceosome is the enzyme that's involved in this cut and paste. And then up at the top, we had RNA polymerase involved in the actual constructing of the mRNA. And then remember that the mRNA is going to leave the nucleus and go to the ribosome for translation. And so just to kind of help simplify, I'm going to rewrite over here my mRNA transcript. So I'm going to have UAC, and I'm going to put them in groups of 3 letters. UCC, AUU, AAU, CGG. So I didn't actually edit mine. I didn't cut or paste anything. So everything that I originally transcribed, I'm putting into this final mRNA. Now, as we look at it, I said, oh, hey, go ahead and write it in groups of 3. So I had UAC as a group, UCC as a group, and so forth. Each of these groupings of 3 we call a codon. So a codon is like, it's kind of like our word. So our A's and U's and C's and G's are sort of like our alphabet, right? And then we can put them together in groups of words, and all of our words are just spelled with 3 letters, and each of those 3 letters is a codon, all right? And these have meaning. This is how we build our protein. So now what we're ready for is our tRNA molecule, So our tRNA molecule, remember, has the job of bringing in the ingredients, and the ingredients here would be the amino acids, right? The building blocks for our proteins. And so what I like to do here is actually sort of like draw my tRNA molecule like it's a stick figure where the hands are holding onto what we would call an anticodon, and then the feet are holding on to the amino acid. So an anticodon is complementary to the codon. Anticodon is complementary to the codon. So if the codon is UAC, the anticodon would be AUG. Right? And that's how that would work. The tRNA then is using this to bring the ingredients and know when to drop them in the bowl, basically, right? And so based on the order of the codon and the matching up with the anticodon, then the amino acid, the correct amino acid will be brought in line. And so what we would do, that's how it works in the cell. But as humans, we don't just pull out the tRNA. Instead, we've sorted all this information out. And so we use what's called a codon chart to figure this out. So let's take a look. Let's interpret an mRNA sequence. Hang on a second. Okay, and so we're going to look at this RNA codon expression table. And so we're looking here at the bottom side. And how do we know that it's RNA? Well, simply all we have to do is notice that we see uracil or U's instead of thymine. So the table up above has T's, and so that's showing DNA, and we're not going to be paying attention to that table. Now I've included this because this is the table that you might see when you do your final exam, and so I wanted you to have a little bit of opportunity to see that. Okay, I'm going to go ahead and rewrite on this screen my mRNA codons that I did before. So you already have this on your papers: AUC, UCC, AUU, AAU, and then the last one is CCG. And I'm running out of room, so let me do it a different direction. AUC, UCC, AUU, AAU, CGG. So I'm not real happy doing a sort of vertical display. I feel like it makes more sense when it's horizontal, but I don't have room for that. Okay, and so what we do is we use our codon expression chart and we look for the codon AUC. So this is adenine uracil cytosine. And when we see that on our chart, we see that it's going to code for the amino acid. So here's mRNA, and here we put our amino acids, which I'll abbreviate little a. And so we have ILE as our first amino acid, and that's just another abbreviation for amino acid here. I don't actually remember that one at this moment. Let's find UCC. Here it is. And so it's coding for amino acid serine, which is abbreviated SER. Let's find AUU. So here we're coding for ILE again. And so we do see some redundancy in our code, which is nice because if we get mistakes in our DNA, we're less likely to have mistakes in our protein. The next codon is AAU. And so we see ASN as that amino acid. And then the last one is CGG in our listing here. And so find that here. And so that's standing for ARG, which is arginine. Okay, so I want to go back to this picture here where we have our mRNA listed in a horizontal direction, and then I have my little tRNA drawn in. And instead of just having the generic amino acid coming in here, we know from our table that it's ILE. And then we can go ahead and fill in here using that information from our table. So hopefully this is how you completed your notes already. And I'm doing these little dashed lines in between to represent peptide bonds. Remember that our primary structure of our protein is an amino acid connected by peptide, amino acids connected by peptide bonds. That's what we see as our beginning protein structure. And this then is basically our transcription and translation. I would like to go back to our table, our chart, just to emphasize something. We do have what's described as a start codon. A start codon. And the start codon is AUG. And it codes for the amino acid methionine. So in real life, the beginning of every gene is going to have that AUG. The beginning of every mRNA transcript is going to have that AUG. And we also have what are called stop codons, which are not actually on this particular table. And a stop codon tells the cell that you're done. That's the end of that gene. So we'll see that. All right, next video up is all about cell division. So as always, take care of yourselves and each other.